Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, I will discuss a very important topic that is related to microbiology. And the name of the topic is the effect of oxygen over microbial growth. As we know that bacteria are such type of microorganism that show different responses to variety of responses to oxygen concentration. There are such type of bacteria and even protists that when oxygen is in an abundant amount, so certain type of bacteria grow well in the high concentration of oxygen, and there are such type of bacteria and protists that cannot grow well in the presence of oxygen. And I want to clear to you people that all the multicellular organisms depend on oxygen. But here the case is just with the bacteria, the protists, and even the fungi. Fungi also show a variety of responses to oxygen concentration. Okay, so in this video tutorial, I will discuss that what are the different classes of microorganisms that are divided or classified on the basis of oxygen concentration. And in the last, I will discuss that why these such type of bacteria shows different responses to oxygen concentration. Means that, for example, there is a bacteria and it can grow well in the presence of oxygen. So I will give the reason that why it is growing well in the presence of oxygen. And the second one, that suppose there is a bacteria and it is not growing well in the presence of oxygen. So I will give the reason that why this such type of bacteria is not growing well in the presence of oxygen. Okay, so our concern is mainly with the reasons, because mostly in the papers and in the why was the examiner asked the main reason that why does this happens. Okay, so first of all, I will classify these terms, and at the last I will give the reasons. Okay, so let's start with the video. First of all, I will use this sample tube in my video lecture. So you must be familiar with it. Okay, this is a tube, and just look here, the blue portion. There it contains a special type of broth that is known as thioglycolate broth. Okay, and the main color of the thioglycolate broth is yellow color. This is the real laboratory test tubes where the thioglycolate is present. Okay, so mainly it is a media, it is a broth, and this is used in the by finding the oxygen concentration. Okay, the to classify the microorganism on the basis of oxygen concentration. So keep this thing in mind, that in the media that is used is known as thioglycolate broth, and this is the molecular structure. Okay, this is the molecular structure of the thioglycolate broth. Secondly, this green color is the microorganism. Okay, so if you see in the later videos this green color, so you will consider it as microorganism. And just keep this thing in mind, that suppose in the at the top of the tube, oxygen concentration is abundant. For example, it is 100%. Okay, and here in the top is oxygen concentration is abundant, and at the last, just we when we move to the depth of the test tube, so here the oxygen concentration will be 0%. So keep this thing in mind. If just here 0%. Okay, sorry, this is 0%. Now, so if you see bacteria here, so you will consider it as that they are in need of oxygen, they are consuming the oxygen. And if you see here the bacteria, the microorganisms, so you will consider it as that they are living in such condition where oxygen is not there. Okay, so just keep this test tube in your mind. And which broth is used? So the broth name is the thioglycolate broth. This is important for the MCQs, so keep this thing in mind. Okay, now we will proceed further. First of all, you should know about that what are aerobes and what are anaerobes. So in simple, aerobes are those microorganisms that can grow well in the presence of oxygen. So if I just write O2 and give a tick, so this means that aerobes are those microorganisms that can grow well in the presence of oxygen. And the second is anaerobes. So anaerobes are the microorganisms that cannot grow well in the presence of oxygen. These are two terms, microbiology terms, that are used for these such type of microorganism. So the cross means that it cannot grow in the presence of oxygen, and aerobes means that it can grow in the presence of oxygen. Okay, so keep these two terms also in your mind. Now I will discuss these five test tubes in separately. Basically, the microorganisms are divided into five categories on the basis of oxygen concentration. So I will discuss that what are obligate, aerobes, and the other four types separately. Okay, so let's proceed. First of all, obligate aerobes. As the name indicates, obligate means compulsory, and aerobes, as I told you, that microorganisms that are that grow in the presence of oxygen. So obligate means that compulsory. And these are the micro sorry, these are the microorganisms that can only grow well in the presence of oxygen. If there is no oxygen, they cannot grow well. They cannot grow well means that they cannot grow. In simple word, if there is no oxygen, they will directly die, and if there is abundant oxygen, so they will grow well. So obligate aerobe, just look here, the microorganisms are present in the top of the tube. So if you are performing a test in the laboratory and you are finding the oxygen concentration, classifying the microorganism on the basis of oxygen concentration. So if the cloudiness is at the top of the fluid, and you see it in a microscope or even in a UV light. So if here is the colony is here, so you will directly say that this colony is basically obligate aerobes. And as I told you that at the top the oxygen concentration is 100%, so here the obligate aerobes will grow well. So, and just look here, the place the tube is empty here. So this means that here it cannot grow here. Just even if the oxygen level is a bit lower, so it cannot grow. So this means that obligate aerobes. Okay, and they gather at the top. So here the main indication is that it gathers at the top. The examples are the micrococcus, that is Pseudomonas, mycobacterium, and most protestant fungi. And keep this thing in mind, that most of the microorganisms are related to this type of group, that mostly the microorganisms are obligate aerobes. They are using the oxygen, and the multicellular organisms are also obligate aerobes, and most of the fungi are obligate aerobes. Just a few fungi are there, for example, Saccharomyces cerevisiae, that are not obligate aerobes. So if someone asks you that in which category most of the microorganism lies, so you will just answer it that they lies in the obligate aerobes. Okay, so keep this thing in your mind. The second is the obligate anaerobes, just opposite of this, just completely opposite of obligate aerobes. This is obligate anaerobes. So these are the microorganisms that can only grow well in the absence of oxygen, because and if there is a little amount of oxygen, so they will be killed. So they are poisoned by the oxygen. And at the last, I will give you the reason that why are they poisoned by oxygen. Okay, so stay tuned with this video and watch this video till the end. So because we are more concerned about the reason, as a microbiologist, we should know about the reason, because mostly it is asked in the exams. Okay, so obligate aerobes, anaerobes are those microorganisms that cannot grow in the absence of presence of oxygen. And if there is a little amount of oxygen, they will be killed by that oxygen. Okay, so just look here, they are present at the bottom of the test tube where the oxygen concentration is at 0%, as I have told you in the earlier part of the video. The examples are Clostridium, Bacterioids, and Methanobacterium, Trypanosomas, agilis, and etc. etc. There are many microorganisms. Okay, So keep this category in your mind. The third one is facultative anaerobes. Now it is a bit slightly changed from it. Facultative means we can say facilitated. Okay, so these facultative anaerobes can grow with or without oxygen because they can metabolize energy aerobically or anaerobically. These are the anaerobes that can grow well in the oxygen, in the presence of oxygen, and it can also grow well in the absence of oxygen. So they are a bit adaptive type and flexible type. So if there is oxygen, so just look here, it can grow here, it can also grow here, it can grow at the bottom of the test tube. So this means that it can grow well in every type of oxygen concentration. So there, these are basically known as facultative anaerobes. Okay, so, and the examples are the E. coli and Enterococcus and Saccharomyces cerevisiae. As I have told you that some of the fungi are facultative anaerobe. So, but keep this thing in mind. If there is oxygen, so they will give preference to oxygen. If there are two conditions that we have a test tube and at the top there is 100% oxygen concentration, at the last there is 0% oxygen concentration, so they will give preference to this oxygen concentration, high oxygen concentration, because I will tell you in the last that why are they these giving preference to the oxygen. Okay, but if there is no oxygen, they can grow well. So these are known as facultative anaerobes, and these are the some of the example of it. Now the fourth one is that microaerophiles. Microaerophiles. Okay, so what are microaerophiles? This is the fourth sorry. Microaerophiles need oxygen because they cannot ferment or respire anaerobically. However, they are poisoned by high concentration of oxygen. Means that they can grow well in the presence of oxygen, but keep this thing in mind, they cannot grow well in abundant oxygen. Okay, they cannot grow well when the oxygen is abundant. So just look here, they are present here, and I want to tell you that they can grow well when the oxygen concentration is 20%. Okay, if it exceeds from 20%, so there will be then an issue, they will be killed by the high concentration of oxygen. So just look here, they are not present here at the top, they are not present here at the bottom, or just here, they are present in the middle. Even it can be present here, they are present in the middle of the tube. So this means that if there is oxygen, so they can grow well, but the case is that oxygen should not be in a high amount. Okay, and I will give the reason at last. Okay, so they gather in the upper part of the test tube but not the very top. Okay, and the examples are Campylobacter and Spirillum volantus and Treponema pallidum. Okay, so these are the microorganisms that are microaerophiles. Okay, now the last one is the aerotolerant. Aerotolerant are basically aerotolerant are those microorganisms that metabolize unlike obligate, and they are not poisoned by oxygen. Okay, they can be found evenly spread throughout the test tube if the oxygen is present in the abundant amount. So they can grow well. If oxygen is present in lesser amount, they can even grow well. And if there is no oxygen, so they can also grow there. So aerotolerant are also a bit type of flexible microorganism, just like the facultative anaerobes. Facultative, just look here to the facultative anaerobe. As I have mentioned in the previous part, facultative anaerobes can also grow in the test tube throughout the test tube, but if there is abundant oxygen, so they will give preference to abundant oxygen. And just look here, they are not giving any preference to the oxygen concentration. If there is high oxygen concentration, so they can grow well. If there is no oxygen, so they can even grow well. And if there is a little amount of oxygen, so or even also they can grow well. Okay, so they are a bit different from the facultative anaerobes. Okay, facultative anaerobes, they give preference to high amount of oxygen, but in the case of aerotolerant, they do not give any preference to the high amount or to the lower amount. They just randomly evenly grow increase their population. Even Sorry for my coughing. So this is the definition of aerotolerant. Now these were the 5 categories of the microorganisms that are classified on the basis of oxygen concentration. Now here we come with the reason that why these microorganisms show different flexibility, different responses to the oxygen concentration. Okay, so this is the main important part of the video. Just look here, if we take obligate aerobes, and as I have told you that it can only grow well in the presence of oxygen. Okay, so you know that what really oxygen play a very important role in the respiration. If you keep the electron transport chain in your mind, so at the last the electrons are accepted by the oxygen. Okay, the electrons are accepted by the oxygen, and what happens if I write in this electron? Okay, so don't judge my writing, I will improve it. Okay, if the electrons are accepted by the oxygen and it is converted into H2O, and by the formation of H2O the respiration is completed. Okay, as you know that the product of respiration is CO2 and H2O. So the oxygen plays an important role in the respiration. If there is oxygen, so the oxygen will accept the electron and will form H2O, and the respiration will be completed. Okay, and if there is no oxygen, so then the respiration will not be completed. So obligate aerobes are basically those microorganisms that are in need of oxygen. If there is no oxygen, so the electron transport chain will not be completed and water will be not formed. And just because of that, it will not complete its electron transport chain, and just because of it, it will die. Okay, so these are the obligate aerobes. They are very in need of oxygen. Secondly, if I give you the example of strict anaerobes, okay, just I have told you obligate anaerobe, so they do not need oxygen. Okay, they do not need oxygen. This means that the electrons that are present, that are traveling in the electrons transport chain, can be accepted by another type of molecule. Just in the case of the if we take the nitrogen, we can say NO2 or NO3. So what will happen? This electron can be accepted by these such type of molecule. So they are not in need of oxygen. So these are known as strict anaerobes. This is the main reason of the obligate aerobes and the strict anaerobe. The second main reason is that as you know that if there is abundant oxygen in the atmosphere, okay, if there is abundant oxygen mainly in the media, so there are such type of proteins, enzymes that there are such type of enzymes that readily provide electrons to the oxygen. Okay, if there is oxygen, abundant oxygen, so there are many electrons in the inside the microorganism. So what happens that these electrons are added to the oxygen, and such superoxide radicals, and even hydrogen peroxide and hydroxy radical, these are formed. Okay, these type of species are formed, and these species are very much corrosive for the microorganism. Okay, these are very much corrosive for the microorganism. But just keep this thing in mind that obligate aerobes can deal with these type of superoxides. Okay, just look here, this is the f in case of obligate aerobes. Just I am telling you, in case of obligate aerobe, the obligate aerobe has such type of enzymes, mean that superoxide dismutase and catalase and peroxidase, that can readily destroy the superoxide and can change it into oxygen and other important substances. So if there is such type of superoxide, so obligate aerobes can easily degrade it and convert it into useful substances. Okay, but in case of strict anaerobes, these are not having these enzymes. These strict anaerobes are not having these enzymes, be it peroxidase and catalase and superoxides. so dismuted. So what really happens when these superoxides accumulate inside the bacterial body, inside the bacterial cell? So these are directly killed by these superoxide because they are not having the enzyme, so they cannot deal with it, and they are killed by these superoxide. And this is also the case with the microaerophiles. Okay, microaerophiles are just having this in case of microaerophile, these are having the enzyme, but they are not complete. Okay, for example, microaerophile only have this catalase. Okay, this catalase of peroxidase, but and that are these are not having superoxide dismutase. So just because of the lack of enzyme, they can be partially killed by these superoxide. If we look to the facultative anaerobes and the aerotolerant anaerobes and the microaerophile, so this is the same case with these. Okay, the enzymes only in case of obligate aerobes, these three enzymes are present. In case of facultative anaerobes, so this not the three but two enzymes will be present, so they will not completely deal with this oxygen. Okay, this harmful radicals. If we look to the aerotolerant, so aerotolerant will have only one enzyme. Okay, so if it can deal with the superoxide but not in a great way, just like the obligate aerobes. In case of strict anaerobes, these are not having any type of enzyme, this superoxide, so they are directly killed by the these superoxide. So these are the main chemical reaction. I have taken it from an authentic book. So these are correct and correct reasons that are related to this oxygen concentration. So I think now you know that why these are divided into 5 categories on the basis of oxygen concentration, and this is just because of the formation of superoxide. Because in the different chemical reactions are taking place inside the bacterial cell and electrons are produced. Okay, so when there is oxygen, and we all know that mostly oxygen is present in the surrounding, so what happens, these as oxygen is very much electronegative, so these electrons are accepted by the oxygen, and just because of it superoxide are formed. In normal case, as I have mentioned, oxygen can, in the electron transport chain, oxygen accept the electron and convert it into H2O. But when the oxygen concentration is high, there are such type of enzymes that add oxygen, that add electrons to the oxygen in a different manner. So these such type of oxides are formed, and they are corrosive. But obligate aerobes can deal with it, and the remaining cannot. The remaining can partially deal with it, and the strict anaerobes can not deal with it because they are not having the total enzyme content. Okay, so these are the main reasons that are just because of it, these are divided into 5 categories. Okay, so hope you have understand the reason. If someone asks you, so you can easily tell him, because this is, I have taken it from an authentic source. Okay, so this was all about the microbial growth and the effect of oxygen concentration. If you have any further questions related to this lecture, so you can ask me in the comment box. I will directly answer you there. Okay, and if you have understand this video, so like this video, share with your friend, and subscribe to my channel. So in order to get more interesting and conceptual videos related to science and microbiology. Thank you very much. God bless you.